Hi, my name is Cara, and this year I researched the siderophore complex in non-tuberculous mycobacteria. And firstly, I want to thank my mentors at National Jewish Health, Dr. Nabi Hassan and Dr. Jennifer Honda, for donating their time and efforts to help me with this project. So I'm looking specifically at mycobacteria, or shortened as NTM, because the threat of lung disease cases caused by NTM is increasing in the United States, and especially here in Hawaii. Most people know about the lung infection called tuberculosis since we get tested for it. However, not many people know about non-tuberculous mycobacteria that can also cause lung disease. Here I have a chest CT scan. On the middle is a healthy lung, but as on the side, this is lungs infected with mycobacteria, and the one on the left is one that is a person who has gotten repeated exposure to it from just like going into a hot tub and having a good time. Next, I would like to show you a picture of the NTM rates compared to the tuberculosis rates in the US. So if we look at the NTM rates on the side, the number of NTM cases can exceed over 58 cases per 100,000 people. Compared to tuberculosis, the national average is only about 2.8 cases per 100,000 people. So NTM is a really big risk or threat that we should be concerned about. And also compared to tuberculosis, which is usually passed on from one person to another, NTMs are everywhere around us. They have been found in your shower heads, your faucets, air conditioning units, and even water faucets. And so whenever you use these appliances, these bacteria are being aerosolized into the air, and you could be breathing them in. But this is, do not worry too much, because there are species of NTM that are associated with causing lung disease, and which I characterized as pathogenic, and species that are not characterized as much as being associated with lung disease, and I characterize that as non-pathogenic. So I wanted to see if there's a distinction genetically between these two groups, to see if there is something that pathogenic NTM had genetically that non-pathogenic NTM didn't have. The gene complex that I looked at or focused on specifically was the siderophore gene complex. So siderophores are molecules that bind to iron in the external environment and bring it back to the bacteria for it to use. And iron is a really key element because the bacteria use it to grow and multiply. So without iron in iron conditions, the bacteria cannot grow. So this is important because in humans, iron is really tightly bound to other molecules. So if the bacteria cannot get that iron, it can't establish itself and infect the human's lungs. So I wanted to point out some important numbers that I thought. There are a little less than 250 known siderophores, but there are only about 3 of them that are found in siderophores. And there are also 10 genes in the certain siderophore that I will introduce next. So pictured here is the protein structure of the siderophore that I'm looking at, which is called mycobactin. And mycobactin is a siderophore that is found in all mycobacteria, so it's a good thing to study. And as I mentioned before, there are 10 genes to make this siderophore. However, not all species of NTM might have all 10 genes. And since each gene contributes to a part of this protein structure, let's say like the blue or red part, if the mycobacteria does not have this gene, it will have an incomplete mycobactin structure and will not be able to acquire the iron it really needs to grow and infest itself. So this is an important topic because there is no cure for NTM lung disease. The only treatment is a multitude of antibiotics the patients can take, but this is also not a long-term solution because NTM become resistant to these antibiotics very easily. So one method people are looking at to tackle NTM disease is attacking its way of growing instead of actually destroying it. So if we target these siderophore genes, the NTM won't be able to grow or acquire iron that it really needs to grow. So the specific purpose of my project is to analyze the genetic variation within these siderophore genes between the two categories of pathogenic and non-pathogenic NTM. And these are the two guiding questions of my project. I wanted to look at is whether there is a difference in the number of genes between the species and how different are the genes within the Mycobacterium complex. And this second question is important because even if pathogenic species have more genes, if the genes are all genetically very similar, there's really no distinguish. So I wanted to talk about next about how I carried out this project. I used bioinformatic analysis, which is basically using computational programs to carry out your work. And my process of this was gathering data, analyzing that data, and then finding out the implications of it. So using a computer and programs to manage your data is really helpful and key when you have very large data sets. For me, I had over 61 strains of NTM, and there are more than 5 genes per species, and each gene is over thousands of nucleotide bases long. So having a computer program to help me organize it and acquire it was really helpful. Next, these are my my results. So on the top, you can see the Mycobacterium genes, and on the side is the NTM species. The green ones are non-pathogenic and the red ones are pathogenic. So if a species had, let's say, MBTA gene, I would color that box yellow. So as you can see already, there are much more Mycobacterium genes in the pathogenic ones compared to the non-pathogenic ones. To confirm this, I did a statistical test to confirm that pathogenic NTM did have a significantly more amount of Mycobacterium genes. And to answer my second question, which is genetic variation, right pictured is the gene sequences of Mycobacterium C, the gene, and I aligned them, which means we're trying to look for commonalities within, and we're also looking at the differences within the same gene. So each color represents a difference in the nucleotide base in the certain position within the gene sequence. And as you can see, there are a lot of colors, so that means the genes are very different from one another. So to recap, pathogenic NTM had more siderophore genes, which means they have a more complete Mycobacterium molecule to acquire iron. Secondly, there is a lot of genetic diversity within the genes, so that means the protein structure of all a similar part of the molecule might not be all the same, and the differences could lead to more efficiency in acquiring the iron. And third, siderophores are important because the bacteria needs the iron to grow. So, yes. So further research for this project could be looking at the real-time efficiency of iron acquisition within the bacteria, so we can monitor how fast the bacteria acquires iron. And we can also look at how the genetic variation that I pointed out earlier affects the protein structure. So when I first started this project, I didn't know anything about siderophores, and I only had a small taste of pathogenicity and bacteria and things. So one skill that I learned was to when tackling reading scientific papers, you have to get past all the jargon, because if you don't get past it, you're never going to learn anything. And there is a lot of jargon in the papers relating to siderophores, so it was really key to just keep going at it. And it took me a while to fully understand my project, but I got to it, for where I'm adequate. Also, another important thing is to understand the importance of your work. I didn't really understand how my work was helping other people or if it even would help other people. So understanding that when I got to that point where I realized my work is important, it really helped me motivate myself to keep going. I would also like to also acknowledge Dr. Chan for helping me in this process. I got lost many times in trying to acquire data and understanding what that data meant. I would also like to thank again Dr. Hassan and Dr. Honda for introducing me to this topic and being patient with me as I learned what bioinformatics was and what siderophores were. Thank you.